CRISPR-Cas9. Bring me a gene encoding for a specific protein.
Alright, we're going to finish off hemoglobin in this video, and we're going to focus on fetal hemoglobin as well as other diseases that are associated with hemoglobin. And so, if you remember from class, we said hemoglobin, especially fetal hemoglobin, does not bind to 2,3-BPG. And so, one of the reasons why this would be the case is just the delivery system of oxygen in a baby as they're developing. But if you remember where 2,3-BPG can go, it goes right into the pore in the middle. And so what those gamma subunits do is they slightly change the conformation of this pore that inhibit 2,3-BPG from actually binding to that region. And so gamma, if you remember, gamma subunits bind oxygen tighter than their alpha subunit counterparts. And this is due to the fact that they need to remove the oxygen from their mother's blood system. And so that's why. But in class, let's go back a couple slides. In class we said you have fetal hemoglobin. Usually we denote it as HbF. So fetal hemoglobin, one of the bad things and one of the things that they tell expectant mothers is to not smoke when pregnant. And so what can happen naturally, and it happens in all of our bodies, we have two hemoglobin. Right? The hemoglobin is normally an Fe plus 2, right? It's in the Fe plus 2 state, allowing it to bind to oxygen and transport it. What can happen is those two hemoglobins with Fe plus 2 can form two what's known as methemoglobin. So methemoglobin, which becomes then oxidized to Fe plus 3. And we know Fe plus 3 cannot bind oxygen, and it can't do any function. Well, how does our normal hemoglobin turn into methemoglobin, or methemoglobin? And one of the ways is through smoking or various other drugs, as well as endogenous oxidants. So things like chemicals, smog, anything else that might come from the exterior that is really good at oxidizing our hemoglobin. So this happens naturally. So how in the world then do we go back from methemoglobin to regular hemoglobin? Well, there is a concurrent reaction. So there's a reaction going on at the same time, which takes NADH, and what we're going to do is we're going to oxidize it to NAD+, and when we do that, we can then reduce our iron back to Fe plus 2, and the thing that does this is NADH, cytochrome B reductase. Now, all of our hemoglobin, whether we're infants or have fetal hemoglobin or adult hemoglobin, this naturally occurs. And so we need to use our NADH, cytochrome B reductase, to reduce it back to Fe plus 2. But the problem with infants and newborns and babies still in the womb is that a newborn has about half the capacity. So about half the capacity of the NADH cytochrome B reductase than does an adult. And so that's why smoking can be so bad, and it usually is bad for infants or for pregnant women to do, is because the babies don't have the capacity to reduce their hemoglobin, which would decrease the amount of oxygen their bodies would be able to take up. And so that's a thing that we naturally need to watch out for with hemoglobin. But we can also start talking about hemoglobin diseases. And so the most familiar, probably, hemoglobin disease we're familiar with is sickle-cell anemia. Sickle-cell anemia is denoted as HBS, S standing for sickle cell, just like F for fetal. Here, S stands for sickle cell. And so it's one single mutation in your hemoglobin affects this or causes this disease. And so what you have is an amino acid position number 6. Right? So amino acid position number 6 is usually found under normal or under deoxy conditions, right? Conditions where there's no oxygen bound to the hemoglobin, which would be your T state. These amino acids are found up here on your beta strands. And so you have a mutation. You can kind of see it here in this picture. They're kind of putting it flat instead of rounded. I'll go ahead and draw a point. So the mutation occurs about right there. Now, normally, you have a glutamate. You have a glutamate at that position. And so, under deoxy conditions, that glutamate would be on the outside. Now, let's think about the R group of glutamate. It's polar, and it's charged. And so if we get two hemoglobin subunits next to each other, those charges are going to repel each other because it's going to be the same amino acid. That's in the normal hemoglobin. But in the sickle cell hemoglobin, we change that glutamate to a valine. Now let's think about a valine. A valine is nonpolar, and it's uncharged, obviously, because it's nonpolar, but it's nonpolar. Nonpolar R groups become sticky. So under deoxy conditions, we now have a sticky R group on the outside of our hemoglobin. And what that can cause is interaction between molecules. And when those molecules interact, again, this is at the T state with low oxygen. When a lot of molecules start forming, then we can start getting strands. And once we start getting strands, then we can start getting crystallization. We can get the fibers that form, which will then affect the shape of our red blood cells to make them more rigid into more of that sickle shape that we're all familiar with. And so what happens, and the reason why sickle cell anemia can be so bad, is that it causes the blood to become trapped in the small capillaries. Because normally red blood cells are kind of rounded, but they're very flexible. And so they're able to line up like pancakes, or coins, if you will, to get through those small capillaries. But there's still a little bit a give to the red blood cells. When you get sickle cell anemia, you now have a sickled cell. And it's a lot less forgiving. It's a lot less malleable. And so what's going to happen is it's going to get stuck. And so what you can have is you can have some crisis that'll occur. And so what also can happen in sickle cell anemia or patients that suffer from sickle cell anemia is they can be anemic. Because you're going to clear out those red blood cells a lot faster. So there's going to be a faster clearance of the breakdown of those red blood cells, causing you to have low red blood counts and be anemic. Now, there is no cure for HBS. So let me go ahead and erase some of this stuff over here. So there's no cure. We can't just give a pill and say, look, you're cured of HBS. So no cure. But with that being said, there are some partial bone marrow transplants that are occurring. There's some genetic gene therapy with a bone marrow transplant that's occurring at the NIH that is very promising. But right now there is no cure. It's a completely recessive trait, and so you need both copies of it, both mutant copies of it, to actually have sickle cell anemia. But, I'll put a but here, but one copy confers malarial resistance. So I'll just do resistance to malaria. And so if you have the disease, if you have sickle cell anemia, you will be resistant to malaria as well, but only one. The heterozygous individual, the person with one mutant copy, will also have resistance to malaria. And so in a way it's bad, right, because of what could happen in your capillaries and some of the issues that can arise, but it also has an advantage to it in that you're resistant to malaria. There is another hemoglobin disease. Let's go to the next slide. There is another hemoglobin disease that's called HBC. So we saw HBS with hemoglobin sickle cell. Here is HBC. The C stands for quote-unquote crystallizes. Right? Usually HBC goes undiagnosed. And then position number 6, so again the same position, you make a glutamate to a lysine. So we're going from charged negative to a charged positive. And so, and depending on the binding, because there is going to be a negative patch on your hemoglobin with that glutamate and the alpha, and so then those will be able to interact. The disease causes mild chronic hemolytic anemia. So again, like what we saw with sickle cell anemia, is that your anemia is going to be an issue. There's no therapy required. It's not as bad as sickle cell. Your blood cells still remain viable and about the same shape. It affects 0.01% of African Americans. And again, HBC, like we saw with HBS, confers resistance to malaria. Now, so we have HBS and we have HBC. We can also have, let's do this in a different color. I'll keep this up, but we'll go and do this next one in a different color. HBSC. So you have both the sickle cell trait and the crystallizing trait. Now, usually this has no problem whatsoever, unless you start seeing problems with this type of disease when you have what's known as infarctive crisis, where you lose lots of blood or you need blood. So things like surgery or childbirth, or you fall and you get a really nasty cut. These are the types of situations where we see HBSC manifest itself in a bad way. And so, but usually HBSC has it never goes, it's very rarely diagnosed. And so those are the hemoglobin diseases, at least that we're familiar with: hemoglobin S, hemoglobin C. There's also diseases called Thalassemias. And what happens is in all of us, our hemoglobin makes a transition from fetal hemoglobin to adult hemoglobin. A lot of times when if we have a disease when one of these thalassemias, it doesn't make that transition. And so there's something called Alpha-Thalassemia so alpha-thalassemia, you have 4 copies of your fetal hemoglobin still there. And so you can make gamma 4 tetramers. Some of them have beta 4 tetramers, right? And so either all 4 gamma or all 4 beta. Because you lose the gamma, the transition is still made, but you still don't make alpha, hence alpha-thalassemia. These are both fatal at the fetal stage. So usually the baby is not born or is born but does not live very long. This is known as HBH. And the problem with these, the problem with having 4 of all the same subunit is there's no cooperativity. Hemoglobin thrives with cooperativity. And if there's no cooperativity, it cannot bind oxygen as well as it should. And so we have alpha-thalassemias, and now we have beta-thalassemias. These only have 2 copies, right? There's only 2 copies that are missing or changed. You know, there is a minor beta-thalassemia, so some of your hemoglobin still gets converted over to the adult kind, some of it doesn't. There's no need for treatment if you have minor beta-thalassemia. Major beta-thalassemia, it manifests with progressive anemia. So as your HbF goes down, you start seeing symptoms, right? So the longer after a baby is born, the more symptoms that will show up. Now in this one, in the beta-thal, so we talked about gamma tetramers and beta tetramers. You can have alpha tetramers. But if you have alpha tetramers, they will precipitate out. And what they require as a treatment is a blood transfusions or a bone marrow transplant to try to get that hemoglobin, the 4 subunits as the correct subunits. And so this ends the chapter on protein function dealing with hemoglobin. If you guys have any questions about what we've covered in these videos, please come and ask me. We do these videos just so I can deliver some more of the content, and so it's very important that you understand the content, that you learn the content. And so if you have any questions, then please come see me.